Dr. Sharma. He received his PhD in life sciences from JNU, New Delhi, working on the regulation of nitrate assimilation in phytochrome. He then conducted postdoctoral research with Dr. Kumar at Yale and at Wayne State University in the areas of DNA-protein interaction. He then did postdoc work with Professor Sapori at JNU and with Professor Hayati, am I saying that right? At the University of Delhi in the area of signal transduction during the regulation of genes and plants. At UDSC, he worked from 1997 to 2023 in the area of structural and functional genomics of tomatoes and in other areas as well. This work included sequencing part of chromosome 5 as a member of the International Tomato Genome Consortium and development of tomato plants with improved nutritional quality and shelf life. Some of his other areas of research include studying the role of methylated DNA binding proteins in gene silencing of Arabidopsis and the development of edible vaccines against cholera via transgenic expression in tomatoes. So Professor Sharma was also nominated as a fellow of the National Academy of Sciences in India in 2012. So please join me in welcoming Dr. Sharma.
Thank you for nice introduction. Is it better or should I go that side?
It's whatever works for you, but there's the camera behind you.
Okay.
The audio input is not set to the microphone.
I don't believe we have a microphone for this one. Yeah.
Okay.
Okay. So I want to introduce this topic. As the title indicates, I'm going to talk about methylglyoxal and glyxalase system in relation to development of tomato fruits. So basically, methylglyoxal is a kind of molecule which is a toxic molecule, and a glyxalase is the enzyme which kind of detoxifies this molecule, and it's not very common to find mention of glyxalase in standard textbooks. So there is hardly any mention, and I want to tell a little bit of history of this enzyme. I did PhD from JNU, and one of my lab mates was working on glyxalase enzyme, and he also got clue for working on glyxalase from his friend who were working in the area of cancer biology. So prior to study in the plants, we knew that this enzyme is having some association with oncogenesis, like in malignant condition, the level of this enzyme goes high. So initial work at JNU, where I did my PhD from, carried out by other people, not by me. So that was in that direction, whether there is any correlation between activity of glyxalase and growth of the cells. So they analyze activity in meristematic tissue, fast growing, slow growing, and they found some correlation of glyxalase activity with the status of cell division. Later on, one of the members of Professor Pori's group purified the enzyme, and they could show that this has similar activity, similar to the one, the mammalian enzyme. Then the gene was cloned, and transgenic approach was used later on. And Professor Pori's group expressed genes for, there are two genes which complete the cycle, and we'll talk about a little later. So basically, they try to express glyxalase 1, glyxalase 2 genes together, and they found that those plants, like they tried in different plants like brassica, rice, and some other plants, and they found that if you express both the enzymes, it provides tolerance against salt stress. So with a little bit of history of this enzyme, I come to the kind of work that we did in our group. So when we talk about the ripening in tomato fruit, we know that ethylene is one of the major regulator of ripening across plants. There are different types of plants. Some have slow rise of ethylene during ripening. In some cases, you have burst of ethylene, which we call climacteric ripening. But there are other hormones like auxin, ABA, lactonic acid, racinosteroid. They also play a role in fruit ripening. And there are some of the less known regulators of fruit ripening. For example, there is a peptide, phosphosulfokain, is 5 amino acid peptide. This has hormonal property. This has a receptor which it binds to. And then the receptor can modify transcription factor. There is TRAP2F, which can phosphorylate, and that affects fruit ripening. So there's another lesser known aspect of fruit ripening is what I'm going to talk about. So this is Methylglyoxal-glyoxalase system, and we try to find out if this system has some role in fruit ripening. So as I mentioned, the glyoxalase, methylglyoxal is not a very popular kind of system to study, but still you find mention about methylglyoxal in Strife's biochemistry book. And what this is showing, glycolysis phosphate step. Glucose is converted to dihydroacetone phosphate and glyceraldehyde 3-phosphate. And this is the enzyme, Strife's phosphate isomerase, which converts dihydroacetone phosphate to glyceraldehyde 3-phosphate. Now, this is the kind of key pathway for generation of methylglyoxal also. So what happens is that is intermediate, and this can be converted to methylglyoxal. And this enzyme has got a loop, this small loop. This kind of blocks the side activity. Main activity we know about what it does in glycolysis, but this side activity is inhibited by this loop. And this reaction, the side activity, if you check in cell-free extract, this is 100 times faster than the physiologically active activity. So basically, what I'm trying to say that when everything works fine, this dihydroxyacetone phosphate will not be converted to methylglyoxal. But when things are not working fine, it may be some kind of stress, which kind of changes the conformation of protein, and that then probably side activity overtakes, it becomes much more prominent, and that is the condition when you can expect much more of formation of methylglyoxal. So methylglyoxal is kind of a devil. It affects many different kind of molecules. It can modify amino acids like arginine, lysine, and cysteine. I made a mistake here. One of the amino acids is cysteine. So this shows how this adduct formation happens. So basically, these amino acids in the proteins like histone or other proteins, transcription factors, these amino acids can be modified so the carbonyl groups are transferred, and that can change the property of these proteins. So that's what, and these products are called this advanced glycation end products. And so this is a problem methylglyoxal can cause. So it can alter the property of different kind of proteins, non-specific. So first, when we said about finding a role of glycolase, we tried to see how many genes are present. So we try to analyze the existing data. So as you can see here, there are, okay, so basically before that I would like to mention there are uh, two activities which are required for conversion of methylglyoxal to non-toxic product, which is lactic acid. So Gly1 and Gly2, these are Glyoxalase 1, Glyoxalase 2. These are the two enzymes which are required to convert methylglyoxal to non-toxic product. There is another kind of enzyme. So these are 15 Gly1 members and 4 of these Gly2 members which are there in tomato genome. And there are 3 members which are in different categories. This is Gly3. So one aspect is that Gly1 and Gly2 form pathways which can convert methylglyoxal to lactic acid. This one, single protein, can convert methylglyoxal to lactic acid. So there are two enzymes system, and there is one enzyme system for conversion of methylglyoxal to non-toxic product. So these are the pathways. So Gly1 can, glutathione binds to methylglyoxal and it forms this intermediate hemi-diacetyl. Then Gly1 works on this, converts to S-lectyl glutathione. Then second enzyme converts this intermediate into lactic. And then another pathway is the one which is catalyzed with Gly3. So here single enzyme can convert methylglyoxal to D-lactate. Now one of the difference in Gly1, Gly2 system and Gly3 system is that Gly1, Gly2 system requires glutathione for it. As I mentioned in the beginning, that glutathione binds to the substrate and then converts this to any. But Gly3 has different pathway. It doesn't require glutathione, and in single step it can convert methylglyoxal to lactic acid. So for finding out whether Glyoxalase enzymes have any role in fruit ripening, first of all, we try to find out of all these things that we characterize, how many of these are showing food-specific expression. We check the profile, and Gly14 is member of Gly1 family. So as you can see, these white bars are for non-transgenic stages. Leaf, mature green activity goes up, breaker it goes up, and then it stays high. This is another member, Gly17, again representing Gly1 enzyme. So this also shows food specific expression. And one of the member of Gly2 family, Gly22, this also shows food specific expression. Now this is one thing, and then we also try to find out whether this somehow interact with known regulators of food therapy. So we use mutations to characterize it. So this first one is wild type alisaccharide. Then there is mutation ring. Next is NOR. Then we have moneymaker, which is the background for SP2 mutation. Then this last but one PR. This is background for the last one. This is the transgenic line where we use RNAi to knock down the expression of ring gene. So what you see here is that wild type is showing increase in the activity, but in these mutants, especially ring and NOR, you see the expression going down. Moneymaker is not so much of effect. And again, when we use transgenic like here, this last one, where we do RNAi for the brain. This is behaving similar to brain mutant. So it shows that probably RIN has some role in, this Glyoxalase has some interaction with the RIN, and when RIN is not present, as you can see, the fruit specific rise in the expression of Glyoxalase gene is blocked when RIN is not working. So these were the fruit specific family members of the gly, which we try to work on. We use this gene, gly14 and gly2 for later studies. So when we set out to find out the role of methylglyoxal, we try to find how does level of methylglyoxal modulate during reaction. And so in the green, this dark shade is for high expression, as you can see in this marker. So level of, if you see wild type, level of methylglyoxal comes down as gradually proceeds. Like in the green stage, you have more, then greater starts going down. But if you see this expression in the ring mutant, you can see in the wild type it's coming down. But if you check the expression in the ring mutant and non-mutant, you see expression is not coming down. So that means methylglyoxal is accumulating because Gly1 enzyme is the one which is going to convert methylglyoxal to non-toxic product. And if the activity of Gly1 is not there, then it will keep accumulating. So that's what happens as tomato grows, methylglyoxal keeps accumulating. It's in much higher amount. If you knock out HP2, there is not so much effect. If you knock down ring by transgenic approach, then also you see accumulation of methylglyoxal. So as I mentioned in the beginning, how methylglyoxal works is one of the ways it forms advanced glycation end product. So we tried to find out whether in our system also, in those conditions when we see high amount of methylglyoxal accumulating, whether there is formation of advanced glycation end product. So this is the detection of glycation end products during Western blotting. So this is the level of end products, glycation end products in the wild type alisaccharide. In the ring mutant, you can see this. You can see more number of band and more intense band. So glycation end product increasing. Same is the case with the non. In moneymaker, this showing pattern similar to wild type. When we knock down ring by RNA approach, again we see more glycation end product. So in a way, showing that there is enhanced accumulation of methylglyoxal, and when methylglyoxal is in high amount, it binds to proteins, different amino acids, and those products can be visualized using the antibody against methylglyoxal in Western blot. Then again we tried, like fruit, when you you see normal fruit ripening. And what we are trying to say is that methylglyoxal blocks this fruit ripening, using mutant approach and RNA approach. So what happens if you try to increase the amount of methylglyoxal? So we injected methylglyoxal and then see whether that can block ripening. So in the wild type, you see a normal kind. This is the normal kind of ripening. If you give 10 millimolar methylglyoxal, you can see there is inhibition of pigmentation. You enhance the concentration to 25 millimolar, you see more drastic loss of the pigment. So this shows that, methylglyoxal, if it is enhanced in its accumulation, its level enhances, then it's going to block the glycation. And in these transgenic lines, we also check the activity of Glyoxalase I and Glyoxalase II. This is the enzyme activity. So this is the wild type. This is again after wild type ring, NOR, then this moneymaker background and mutant, and last RNA protein. So you can see here that there's not so much of difference here in the mature gene stage, but when you try to analyze the activity of the Glyoxalase in ring mutant, you see there is heavy loss of activity. Breaker stage, breaker plus 8 days stage, both these stages, ring and NOR, they behave in similar way. Both block the accumulation of Glyoxalase activity, and same as the pattern for Glyoxalase II. This is enzyme activity, not transcription. So same kind of pattern is also seen. So that means when you have deletion in ring, somehow it affects Glyoxalase activity at the level of gene expression, at the level of protein activity, enzyme activity. So we try to analyze the promoter of Glyoxalase gene, this Gly14, and we look for the cis-acting elements in the promoter. And what you see of interest is CARG box. CARG box is the site where ring binds. So many of the ripening related genes, they have CARG box in the cis-acting elements, and ring binds to promoter of these ripening specific genes. So this was of interest. Then there is also cis-acting element specific to ethylene biosynthesis, which is found in genes like ACC synthase and other ethylene responsive genes. So what we set out, we checked, since there is ethylene responsive element, we checked whether ethylene can enhance the activity of Glyoxalase. So we provided ethylene treatment, and this shows the activity increasing. This is after 1 hour, this is after 3 hours. And we compared with standard ethylene responsive gene, which is E8. E8 is the one which is kind of a model for ethylene responsive gene. And this promoter has been used for driving any gene in ripening specific manner in a number of studies. So this is one of the well-characterized food specific promoter. So that means it's behaving in similar way. I mean, Glyoxalase also has ethylene responsive element. E8 also has got. So both are showing ethylene response. Like in response to ethylene, the transcript level goes up. Now here we tried inhibitor of ethylene. You can see when you use inhibitor of ethylene, it affects both ethylene responsive promoters of Gly1 as well as E8. Again shows that there is some ethylene responsive element in Glyoxalase I, so it is ethylene responsive. And then we also checked, as I mentioned, there is this element CARG box where ring binds. Ring is a MATCOX transcription factor. There are many other MATCOX transcription factors which have role in fruit ripening, and they can form homodimer, they can form heterodimer. So ring is in that class. So we wanted to check whether ring binds to promoter of Glyoxalase. We found the ring specific element in Glyoxalase CARG box. So we tried to find that by different ways. So one of the way we use these two hybrids. So we had construct for ring and Gly4, and ring has been reported already. Like in the literature, we know that RIN binds to the promoter of AC synthase. So this we use as a positive control, and we could find that the interaction between RIN and Gly1 promoter in these one hybrid assays, and these are the negative controls. Then we also tried in other ways, and here we are using co-infiltration of two constructs, one reporter, one effector. So reporter construct is expressing GFP under the control of Glyoxalase promoter, and the effector one is where 35S promoter is driving the expression of it. So you can see when you have RIN alone or mock experiment, or you have just the Gly promoter driving GFP, you have less fluorescence. When effector molecule is there, RIN is driving the expression of GFP through promoter of Glyoxalase I, and you can see much more fluorescence. You can see quantification of fluorescence here. So basically, this again indicates there is interaction between RIN, the max of transcription factor, and the promoter of Gly1. Same thing we also characterize by mobility shift experiments. So we use the CARD box sequence for gel retardation, and you can see that is shift in the mobility of the probe. This again indicates that RIN binds to Glyoxalase I promoter, where the CARD box is there. So by using three different approaches of each one hybrid and co-filtration, and by mobility shift experiment, we can say definitely that there is a site in the promoter of Glyoxalase I for binding of RIN transcription factor, and physically in vitro and by in vivo experiments, we can emphasize on this point that there is interaction between the RIN and the promoter of Glyoxalase I. So when we establish that there is interaction between RIN and Glyoxalase I, then we wanted to see the role of Glyoxalase in ripening, and we tried to silence Glyoxalase by two approaches. We use two different kind of promoters. So in one of the constructs, this one. Okay. So we use a microRNA based silencing strategy here, and with promoter that we use two different kind of promoter. In one of the construct which indicates RIP here, this is for ripening induced protein. Like when we were working on other projects, we wanted to overexpress certain genes for antigen at high level in tomatoes, and we were looking for promoters. So we tried to characterize the promoter, and we checked EST's expression, and we got to one of the EST which was enhanced very high degree in food specific stages. So we isolated the promoter of that, and we call ripening induced protein promoter. So this promoter behaves not exactly similar to E8. Like if you see the expression of E8, you see hardly any expression in green fruit. At the breaker stage, you see significant expression. Then in red ripe, it goes further. But for this one, RIP, what we call RIP promoter, it has very little expression at the breaker stage also. But at red ripe stage, it goes very low. So if you want to have expression in strict food specific manner, it's works better than other promoters. So one of the promoters that we use is RIP. So this is fruit specific promoter. As I mentioned, expression is very little in the breaker stage and green fruit stage and very high in red ripe stage. Another promoter we use substitutive promoter, 35S promoter. to drive the expression of this microRNA-based silencing construct. So now you can see what happens. So this is normal ripening. When we silence Glyoxalase by using ripening specific silencing, you can see hardly any effect till breaker stage, and then towards late ripening stage, you see this green is not getting converted to red, and kind of you have slight yellowish orangish color develop. So there is significant silencing if you compare with the wild type. So red pigment is not developing if you silence Glyoxalase I gene by using ripening specific silencing. Now here in this case we use QT promoter for driving microRNA construct for silencing. Now here you see not just the ripening, but development of tomato fruit was also compromised, and we can understand why. Because F35 promoter would start working right from the beginning, so even at the early stage of tomato fruit development. So that's where also it's affecting. So as a result, fruits are not able to mature to the perfect size, and pigment development is also compromised. So there is no red color development. This is showing much stronger inhibition. Yeah.
So a couple conduits more like a glyoxal.
Yeah, see, that's why I'm saying that Glyoxalase enzyme basically metabolizes methylglyoxal. So if you kind of knock down this enzyme, then there would be accumulation of methylglyoxal. So in this case, there is inhibition of the accumulation of inhibition of Glyoxalase enzyme. So more methylglyoxal is accumulated in this case, because we are silencing Glyoxalase I enzyme, and this is the enzyme which would neutralize methylglyoxal if you convert to non-toxic product. So that means when you inhibit the gly activity, there is increase in methylglyoxal, which is not allowing ripening. But this silencing is happening in food specific. Okay. So here it's not happening, here it's not happening, here it starts, because our promoter here is ripening specific promoter.
Yes.
Okay. But here, when we are using 35 promoter, even before any flower develops, it will start working. So like silencing would happen from the very early stage. So from the level of initiation of flowering and then setting of fruit, everything is affected. So in this case, because Glyoxalase is normal till this stage, when we are using this kind of construct, so fruits are able to develop normally, because in those early stages, there is no compromise happening for Glyoxalase activity. But when we are using 35 promoters, even at the early stages of development of food, you have created a situation where enzyme is not working, and so methylglyoxal is accumulating. It keeps accumulating, more and more is getting system.
So the leaf or the stem goes out.
Yeah, they will also. Yeah. And another thing, as I mentioned in the beginning, the way methylglyoxal is synthesized, it's basically from this step where dihydroxyacetone phosphate is converted to glyceraldehyde 3-phosphate. So what are the situations where you have more, like in the literature there is information that there is a strong correlation of the level of methylglyoxal with the presence of glyceraldehyde 3-phosphate. And this glyceraldehyde 3-phosphate comes from two sources. One is glycolysis, from photosynthesis also. So green tissues would have more of glyceraldehyde 3-phosphate, and they keep getting interconverted, like dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. So there's a correlation in those stages where you have more of the aldehyde 3-phosphate. So green fruit, I mean something like green leaf. Now, as I mentioned in the beginning, green fruits have got more methylglyoxal compared to red fruit. Because photosynthesis is still active, and there's glyceraldehyde 3-phosphate, which is synthesized, and phosphate are synthesized during photosynthesis. So that also, contributes. But for your question, why small size is there? Because, methylglyoxal got enhanced because of knocking down of glycates. The glycate enzyme was not there, so it's accumulated. So right from the beginning of development, it affected in this case. So then, as I'm contemplating, when you have inhibition of glycase gene, there is more methylglyoxal. So this again, we look for glycation products. If you have more methylglyoxal, you expect to bind to amino acids in different proteins. So we saw the products. So this is in the Y type, mature green, Baker +4, Baker +8. And now if you see when we block the transcription of Gly1 gene, you see much more intense products here. Because this is, as I mentioned, we use RIP promoter and 35 promoter, and RIP is not very active in early stages of development. So if you see these stages, Baker +4, Baker +8, you see there is enhanced level of accumulation of glycation product. So this also indicates, and when we compare with the 35s, you see more severe, defect in fruits when you use 35 promoters, because there is stronger silencing of Gly1 gene. So you have less accumulation of Gly1 enzyme. As a result, you have much more accumulation of glycation product when you use 35 promoter driven silencing. So this again indicates that when you have a situation where you block the activity of glycolase enzyme by knocking down the expression, then what this is having, what kind of implication this have is that methylglyoxal keeps accumulating because it's not getting converted to lactic acid because of lack of glycolase. So in these different transgenic lines, we checked kind of oxidative stress and methylglyoxal level. So if you see, like this is the level of methylglyoxal in lower one is wild type. So it's low. Now, when you silence glycolase by using ripening specific silencing. So this one lower, from bottom, second and third. This is silencing of glycolase expression by using ripening specific promoter, which is like moderate kind of silencing. And here we are using 35 promoter driven silencing of glycolase, which is much stronger. So you see, when silencing of glycolase is stronger, you have more accumulation of methylglyoxal. You have more accumulation of the product of oxidradicals like lipid peroxidation. So malonaldehyde content goes up. These are the markers of oxidative stress. So these are going up. So same is the case in green fruit, same is the case in Baker, and all these stages where you have silencing of glycolase. Stronger is the silencing. This last two, there are two lines for each construct. So like this is where 35 promoter is being used for silencing of glycolase, and these two where the promoter is being used, and this is glycolase. So you can see increase in methylglyoxal increase in methylglyoxalase content. And when you have oxidative stress, glutathione is the one which is used by the cells to neutralize those free oxygen radicals. So we also look for the content of glutathione. So this is in the, you can see in the different stages of fruit, like mature, green. So you have high amount of glutathione. In those constructs where we use ripening specific promoter to drive the silencing, you see almost similar level because this is green fruit and this ripening specific promoter doesn't do anything in the green state. So again you see, but when you use 35 promoter, this is knocking down the expression of glyc, and you see glutathione level coming down. At the breaker stage, this promoter starts working. So at the breaker stage, if you see, then even when you are using ripening specific promoter, then also level of glutathione is coming down, and it's much more severe when you use it 35. We also check the ratio of glutathione reduced to oxidized. So obviously, when you have oxidative stress, reduced glutathione will be used up. So this ratio of reduced to oxidized changes. And again, you can see that let's go to these ripening specific stages. This is in wild type, it's much higher. Then in the silencing, where you're using ripening specific promoter, it's decreasing. And when we are using constitutive promoter, it's reducing. So there is correlation between the activity of glycates and glutathione, methylglyoxal, malonaldehyde. We also check the levels of antioxidants, total antioxidants, and you can see as food ripening progresses, there is increase up to breaker plus 4 stage. But when we silence glyc expression, then you can see antioxidant level is coming down. Whether we're using fruit specific promoter or we are using constitutive promoter, level of antioxidants is coming down. So kind of methylglyoxal, when it accumulates, we can say that it's kind of leading to oxidative stress directly and indirectly via change in the concentration of glutathione, other antioxidants. So we check the level of pigments and the expression of genes involved in regulating pigment development. So you can see, so in the mature green, all these stages it's similar because as the ripening progresses, there is increase in lycopene. You can see slight initiation at the breaker stage, and then breaker plus 4, breaker plus 8, and 15 keep going up during ripening. This is the wild type, and then in other plants, like first is wild type, then next two are silencing by ripening specific promoter, or last two by constitutive. So you can see when we silence glycates more strongly, then level of lycopene is going down, level of beta carotene is going up. We also check the expression of genes involved in biosynthesis of these pigments. So you can see phytolene synthase level in the wild type. First line is wild type, and this is the amendment wild type, then non-amendment. So lighter color indicating old level. So you can see level of phytolene synthase transcript going up, level of hydrogen desaturase going up, beta carotene desaturase going up, this carotenoid isomerase going up. This is the white end. But when we have silencing of black phase, we can see that this increase is compromised. You have very low expression of these things, all of these things. And then if we check the expression of genes involved in formation of beta carotene or alpha carotene, you can see the change is different. So that means probably these who are working, I mean, these all the genes, they are working different way compared to these two genes, lycopene beta synthase and lycopene epsilon. These are increasing, but these are decreasing. So maybe these are going up in ring independent manner, and these are going up in independent manner. I mean, we know there is interaction between glycates and ring. So these are affected in different way, but these are probably going up without involvement of ring or ethylene independent manner. So these are not affected by changing the level of glycates, or no, in a way they are kind of stimulated. So which is reflected here in increase in beta carotene. So lycopene is going down, but beta carotene is going up because probably they are following. We check the firmness of fruits. So this is the wild type, white sector. And then when we silence using ripening specific promoter or constitutive promoter, so this is in the green food state. And you can see if you follow this white sector, it is going down, breaker plus 4, breaker plus 8, breaker plus 15. That means firmness is decreasing. This is the normal fruit ripening. But when we silence glycates, let us see breaker plus 4. And you compare with this. This is breaker plus 4 for ripening specific silencing. Now the sectors have increased in size. These two, and this is for 35 driven silencing. Again, they're going further. So basically, when we silence glycates, ripening is getting inhibited because of accumulation of methylglyoxal. So firmness is when you have normal activity of glycates, normal kind of ripening happens. So foods soften in a normal way, but when you block the activity of glycates, then methylglyoxal accumulates, it blocks the ripening, so fruits are firmer. Something similar to ring mutant. There also, when you have no activity of ring, fruits are very firm, they don't develop much color. So here it shows that these lines where glycates is knocked down, they are less firm. Sorry, they are more firm. They are more firm. So this food architecture or texture, is controlled by these lytic enzymes, which break down like pectins and different components of pectin. So there are enzymes that are involved, like pectin lyase. So we check because opening is getting altered. We check the activity of these pectin lyase. So you can see in the wild type pectin lyase activity is much more, but when we block the activity of glycates, this activity reduces. Pectin content also protopectin concentration is increased when you silence glycates. solvent pectin decrease. And this is also, correlated with the expression of different genes involved in metabolism of cell walls membrane. You can see expression of pectin lies. The normal ripening, it goes up at the greater stage and then starts coming down. But when we knock down Glyoxalase, normal ripening doesn't happen, and you can see there is a decrease in the expression of pectin lies. Similar trend is followed by polyigalacturonase. This also normally going up, but when you silence Glyoxalase, then this activity is coming down. Same is the case with pectin esterase, and this is beta galactosidase. So all these genes which are involved in maturation of tomato food and which are involved in softening of the foods, they are compromised when you have increase in methylglyoxal level. So when you silence Glyoxalase enzyme, there is increase in methylglyoxal level, and that somehow is blocking the ripening process. So normal ripening is not happening. So we also check the level of major regulators of food ripening in those conditions where methylglyoxal is accumulating more because of knocking down Glyoxalase enzymes. How is the activity? How is the transcript level controlled in those situations? You can see expression of RIN. Sorry, yeah, this is expression of RIN. It's going down in specific manner, but when you silence Glyoxalase in either way by specific silencing or constitutive silencing, then level of RIN is going down. Level of CNR is also going down. Level of NOR is also going down. And this is tomato agamous-like transcript. Normally these are going up, but when you silence Glyoxalase, then this is also going down. This is the expression of fruitful 1 and fruitful 2. These are the, again, maps of transcription factor which interact with RIN. They also affect some aspects of ripening. Their level is also going down. So basically, some kind of master regulator probably, which is affecting several aspects of ripening is getting affected when you have a situation where plants are not able to metabolize methylglyoxal, and this is the stage caused because of silencing of Glyoxalase gene, and that is leading to low expression of gene, low activity of Glyoxalase. It's not able to metabolize methylglyoxal, and that is affecting most of the ripening specific indicators. These are all transcription factors, major transcription factors which have role in ripening. So now the question is, what happens when we have, we know that ethylene responses are components which are development of pigment, development of softening parameters. So we try to find out where is the effect. I mean, so we know that for ethylene synthesis, the one of the kind of metabolite which ultimately leads to production of ethylene, methane, which is getting converted to SAM and then ultimately to ACC complete. So what we did, we checked the level of aspartate phosphate amino acids, because this is also part of that phosphate. So we check the level of methionine in different stages of ripening. So you see here, these open bars are for the wild type. And so when you have silencing of Glyoxalase, mild silencing, ripening specific, and constitutive silencing, strong effect. So you can see methionine level is going down when you knock down Glyoxalase. aspartate is not showing much change. There is some change in threonine also, but not as much for methionine. Isoleucine is not showing very high amount of change, and lysine. So our interest focus on methionine because it shows some change. So is it that methionine level is getting compromised when we silence glycolase gene? Okay, before like we also check the level of ethylene in these situations when we block glycolase activity. So this is the wild type level, top line, and then when we block is like glycolase gene by ripening-specific acid or constitutive, you can see it's coming down. So ethylene level comes down. So again, the question is where is the disease? Like so we tried methionine. So you can see here when we give methionine to wild type, there is some promotion of ripening. But when we try this in those fruits where glycolase is knocked down, so we add methionine and then observe after 2 days or 4 days, you can see methionine is not able to initiate right. Same is the case where we have silencing, stronger silencing like constitutive. But we tried ACC also. So here when we give ACC, this is in the Y type. So you can see, I mean, you have to compare this with the green state because we use this treatment for green. So you can see that it is improving pigment development. So when you give ACC, there is increase in pigment development. Then you have weaker silencing of glycolase, and also when you have stronger silencing of glycolase, you can see almost no orange pigment, red pigment. Here you see red pigment. So that means methionine is not able to initiate that thing, but ACC does. So that means this indicates that a step before ACC, like which affects ACC synthesis, is blocked when we knock down glycolase. So this shows, I mean, this you are seeing just the pigment, the morphology part. Then we also check whether these treatments, which are causing this change in pigment development, these are associated with increasing ethylene. Whether whatever we are giving methionine or ACC, whether that is increasing ethylene. So this is for upper one is for methionine treatment. You can see at the Y type stage, you can see some difference as I indicated here. But when we use these transgenic lines where glycolase is knocked down, you don't see methionine doing much. No, there's not much increase in ethylene. This is for ACC. So when we are giving ACC, it is working in all conditions, whether you have normal wild type plants or you have plants where glycolase genes are knocked down, ripening specific or constitutive. So this again indicates that probably the key effect is somewhere before ACC is synthesized. So ACC is not synthesized in this situation, and so when you supplement ACC, they start producing ethylene, and you see change in color. So when you have knocking down of glycolase, you have increase in methylglyoxal. So methylglyoxal is affecting some steps, and we know how it can affect. It can form advanced glycation products. So in some cases, it can affect those sites in the enzymes that are related with the activity. Depending on the protein sector, some sites may be exposed where because this is specific to certain amino acids. So certain amino acids in one particular protein may be exposed a little more, So those proteins are going to get more affected by increasing methylglyoxal. So methylglyoxal is somewhere affecting some part of pathway which is responsible for ACC generation. So we also check the expression of genes related to ACC synthesis. So when we give methionine treatment, you can see the expression of ACC synthase not changing. Expression of ACC synthase 4 is also not changing. But when we give ACC treatment, you can see that this is controlled. When we give ACC treatment, then there is increase in ACC synthase. So this again indicates that if you provide ACC, then ethylene biosynthesis is not normal, and somehow methylglyoxal must be blocking a step in generation of ACC. So this is the model that we are giving, how glycolase system works. So this is the wild type situation where a ring binds to glycolase. So as a result, ring can enhance the expression of glycolase, and so in the green state you have more level of methylglyoxal, which inhibits ripening of tomato fruits. But as ripening proceeds, as I mentioned, Glyoxalase I is expressing in food specific manner. So as the ripening stages proceed, and you have increase in the activity of Glyoxalase I gene and the enzyme activity in return, that metabolizes methylglyoxal. So methylglyoxal level goes down, and as I mentioned, when methylglyoxal level goes down, then only ripening happens. So when you have, so this is the normal state. So when we knock down the expression of glycolase, then what happens is that glycolase is not able to metabolize methylglyoxal. Methylglyoxal keeps accumulating, and as we saw, where is the effect of methylglyoxal? Possibly at the level of somewhere in between, like methionine and SAM, and we have strong, evidence which indicates that ACC synthesis is blocked because of high accumulation of methylglyoxal. So when you have a situation where you knock down the activity of methylglyoxalase gene, you have no conversion of methylglyoxal to non-toxic product, lactic acid. So this accumulates, and when this accumulates, it blocks ethylene synthesis pathway in between these steps, and we have evidence which indicates that ACC synthesis is blocked. So it will supplement ACC here, then again this pathway goes normal. So the take-home lesson is that by using different approaches, like you can provide methylglyoxal artificially, that will block ripening. You somehow increase the level, endogenous level of methylglyoxal by blocking glycolase enzyme. Then also methylglyoxal would accumulate. So in any situation where there is high level of accumulation of methylglyoxal, ripening is affected. And we know how methylglyoxal can affect different proteins. It can form advanced glycation products, different amino acids like arginine, lysine, cysteine are the target for, and this methylglyoxal can form glycation product not only with the protein, like proteins, it is known to affect many histone protein expression, or they modify. It also modifies nucleic acid like RNA and DNA, and especially guanine is the target for this advanced glycation product. So this is a kind of molecule which is affecting so many different pathways. And so when you have increase in methylglyoxal, and in terms of protein, we know which are the amino acids where glycation products can be found. But if in some protein, those amino acids are exposed and they get, they provide sites for binding of methylglyoxal, then there are chances that that particular protein can be affected. So in this case, some protein which is involved in ACC synthesis is getting affected by methylglyoxal. And so if you increase the level of glycolase, then it will metabolize methylglyoxal and normal kind of or enhanced fattening can happen. So these are the people who contributed. So all four had been my PhD students, and they are doing postdoc now. So she's in India. Advait is staying here, and she is the postdoc. She was PhD student of my mentor, Professor Pori. So when she joined my lab, and she wanted to find a role of glycolase in ripening, she joined as a student with independent grant. So then we started this work, and he's also my PhD student now postdoc. He did bioinformatics work, and these are my collaborators. Rahul is also my PhD student now in the University of Hyderabad. So we collaborated, and he's my mentor, Professor Puri. As I mentioned in the history of this enzyme, he is the one who contributed in finding out glycolase enzyme in plants, then cloning the gene, then doing overexpression in different crops, and he found that overexpression of glycolase can alleviate different kind of stresses like salt stress and other stresses. So thank you for your kind attention.
We're a little bit over time, so we can continue this. If y'all want to ask questions, maybe in the hallway, we have some stuff back there, so we can continue the conversation afterwards. I do want to present him with this, a little token of our appreciation. So if y'all want to give him another round of applause. Thank you.
You want to ask anything?
Yeah, so increasingly your methylglyoxal is being seen as a signal, not just a byproduct of metabolism. And so whereas a lot is bad, there's some evidence to suggest that a little is good and important for improved root growth under different types of stresses. I was just wondering, in your model, what would happen? I mean, I guess you've already done that if you actually reduced to a large amount of the amount of methylglyoxal that's not made, so you wouldn't have that signal going on. Is it susceptible to biotic stress?
Yeah, see, I have not done, but like other they have done. And one of the way to increase the level of methylglyoxal is by increasing the enzyme.
And what does that do to fruit production? What does that do to fruit production?
Okay.
So now you've reduced the amount, and maybe if you get below a certain level, then is that problematic? That's my question.
See, we also did some work from this stress point of view, and so tomato plants which were very high in activity of glycolase, they provide tolerance to salt stress and drought stress, and so their methylglyoxal levels came down. In terms of fruit morphology, we didn't see much effects in methylglyoxal levels. So probably till certain level, not affecting. So like when you have very low methylglyoxal, probably very less oxidative stress. But in green food, anyway, you have higher amount of methylglyoxal. So I mean, they mature normally in terms of size. So when we, as I mentioned, when we increase the activity of glycates, then we try in different ways. Like we had transgenic lines where glycase 1 and 2 were overexpressed, and we had lines where like 3 was overexpressed. Single line. In both the cases, when we access glycate 3, that provided tolerance to stress, abiotic stress, that we said. But food morphology or pigment was not.
I wonder about fruit chemistry though. You're changing the response to stress. I wonder if it's also changing fruit chemistry.
Yeah, it might change, but apparently, like in terms of the parameters that we have measured, like carotene level, lycopene level, there was not much difference in the overexpressed levels.
So I take this as a, so you have a toxic metabolite that is toxic at any stage of the development of growth. You drop it at the vegetative stage. It will,
Beyond certain amount, that's getting
so it's nearly equal to spraying herbicide. So if you spray the herbicide late in the fruit ripening stage, then you will drop the fruit ripening because when you spray the glyphosate to the tomato, ripening tomato, then it will stop the ripening process at that moment. So where do you think the specificity of this metabolite is ripening specific? Like it's a little bit related that you just mentioned the decreasing the glyphosate doesn't really affect the ripening process. What you said.
Yeah, but increasing definitely affects. If you increase methylglyoxal level, it will affect.
No, decrease.
Yeah, decreasing is beyond level, beyond. So as I'm saying, beyond certain level, because there's always some activity which metabolizing methylglyoxal. So when balance goes, haywire and there is accumulation of methylglyoxal above certain level,
So what triggers that? Do you think there's an imbalance during the ripening process, or imbalance of methylglyoxal during the ripening process, unless you specifically inhibit this metabolite enzyme, there's really no imbalance.
what is getting changed during ripening? As I said, if you don't take any other contribution, and if you just focus on methylglyoxal, as I mentioned, in green stages, where you have more amount of SLDI3 phosphate or dihydroxysteroid phosphate, that could be more in green stage. We have more amount of. So when ripening happens, as I mentioned, this thing that we talked about, like this one sheet, this is ripening specific. So when ripening happens, then you have increase in glycation activity. If this increase didn't happen, it will keep accumulating over the long period of time, and the concentration becomes high if it's not getting metabolized. So that's why we are.
I got it. So what the reason for that increase of the methylglyoxal in the ripening?
Yeah, see, as I mentioned, this glycates has binding glycates motor has binding sites for.
No, the metabolite, not the enzyme. Why it has to? Why the methylglyoxal should be produced more during the time?
Well, it's not produced more. During ripening, it comes down. This level comes down. So when you block glycates, then there is no way to convert this to end product that we can. So when you block with enzyme, you have glycates, then it's not getting metabolized. So that's why it keeps accumulating.
So ripening stage is more sensitive to the normal level of the methylglyoxal. That's the hypothesis. If you keep maintaining the green level of methylglyoxal, it will prevent the ripening. Therefore, you need to produce more of the detoxifying enzyme.
Yes. Because this glycolase promoter is expressing in glycated cells. So when you have this stage, like greater stage, and where other factors like zinc, as I mentioned, they both in the. So brain also shows ripening expression, and black space is controlled by brain. So when you have ripening stage, particular stage where brain starts going up, so that also enhances the activity of glycolate by binding to the promoter of glycolate, and that basically metabolizes the glycolate. So it lends. So if by artificial means also you increase the level of methyl glycol, and you may have everything going like less active, inactive, but if you inject methyl glycol in normal white type food, it will block.
We're recording this on the cloud, right?
I believe so. Yep, recording. Yep.